Okay, hi everyone. This is the last slot of the day. Please give your attention to Yingxin Lin, who's University of Sydney.
Recently moved to Yale.
Okay, so now at Yale University, who's going to be talking about some packages for single cell analysis.
Thank you. So I will. Hi everyone. Thanks for coming for the last session in the last day. I hope that I will finish the workshop in time and hopefully it will be earlier. And yes. So today I will introduce one of the bioconductor package that I have developed. Actually is this is the second version of scMerge, and the first version actually was developed in 2018, which is my first PhD project. And now I have graduated from the University of Sydney and have recently moved to Yale University. So first, I would like to acknowledge people, the co-authors of this package. So my PhD supervisor, Professor Yingxin and Yue, Elijah, and also Ellis here. And I would like to acknowledge the all members of Sydney Precision Data Science Center as well. So we already heard about a lot of single cell data in this conference already. And what I would like to bring about is that how the data structure has been like changing across the years. And in 2023 now, we actually have a lot of like single cell data that are from multiple conditions and multiple sample experiments. So compared to the bulk analysis, when we only have a gene by sample matrix for one conditions, we actually have a collection of matrices from each of the conditions for the single cell data set. And this data structure can be even more complicated if we have two studies that are having similar data structure. Then one of the questions will be how can we integratively analyze this kind of like multiple condition, multiple sample, single cell data set from multiple studies. So we do have like kind of like data that exists in this kind of like with this kind of like a data structure. So one of these is that we have curated 20 COVID-19 PBMC data sets that have around 5 million of cells and 1,000 of individuals. So we have curated the disease severity information as well as like the outcome or age. And then we also and then we curated the expression matrix. So
Can you? It's here, but online they say they're having a hard time hearing you.
Okay.
Yeah. Yeah. If you get a little closer,
I'll ask him if it's better. There's a number of people online.
Okay. And so the question is that how we integrate, analyze the data generated from different experiments. So I will briefly talk about the challenges in this kind of the large-scale integration of multiple studies that compare to just integrate the data that with our multiple conditions. So the first is that now we actually have a multi-layer of unwanted variations. So apart from like the variations that from different studies, like the protocol, sequencing depth, and like different sample handling and so on, we actually also have like that the within each of the study we could have like the batches across the donors and individuals. And secondly is that the biological variation is also multi-layer. So we will have information that associated with the samples, for example, the developmental time or treatment. And also we will have the information related to the cells. And lastly is the scalability of the algorithm. And so, as I just mentioned, this work is actually based on an older version of scMerge. So this is actually based on a model that called remove unwanted variation models. And this was originally developed for the bulk data. So basically, we try to model the data we observe using two main components. So X beta is to model the wanted variation, and W alpha is trying to model the unwanted variations. And the aim here is actually we try to estimate W alpha from the data set using some information, and then we can adjust the data we observe by subtract the W alpha from the observed data set. And as it originally actually developed for bulk data, so it actually requires two information. So one is that we need negative control genes, and then it also needs the replicates. So replicate is quite common in the bulk data experiment. So we can actually, for each of the sample, we can generate replicates, and that would help us to adjust some batch effect or other unwanted variation. But this is not so common in single cell. So the idea here that we create the pseudo replicates. So the replicates is that we're trying to consider the cells from the same cell type as one replicate, and we try to match the same cell type from different batches. So what the procedure here actually is similar to the mutual nearest neighbors cells, but instead of doing it on the cell level, here we actually do it on the cluster level. So the first step is that we want to constrain within each of the batch. And then for any of the two batches within the data set, we are trying to calculate the nearest cluster pairs. So here we're trying to find the nearest cluster pairs for, like, for example, here we find the red and blue are the cluster pairs across the batches. And then we do it for all pairs of the batches. So now we consider like this blue graph as a set of replicates. And we identify all these subgraphs, like cluster graphs, for within our data set, and we treat them as the pseudo replicate set 1, 2, 3, 4. And this will help us to construct the replicate matrix. And this information can help us to estimate kind of the what is the unwanted variation in the data set. And so scMerge have provided two versions. One is that if you don't know any cell type information within each of the batch, that is fine. We will just perform graph clustering. But if you do have some prior information, for example, I know this clustering method works the best for my data set, or I have some public reference data set, I want to use this information to annotate my cell types. So we can actually still use this information as input. And what we call it as a semi-supervised version of scMerge. And this version, sometimes it will be working better than the default clustering method, because sometimes using reference can actually annotate the rare cells better than using clustering to identify the rare cell types. So it will help in the mutual nearest cluster matching step. And another part is that we need to using the negative controls to estimate the unwanted variations. And in this this is the features that we think the expression level are not affected by the factors of interest, and they are stably expressed across different cell types or developmental stages. So basically, if we are having some smart-seq data, it will be like a unimodal distribution. And we have a framework to identify such of the unimodal genes in the smart-seq data. And we utilize this list of genes, usually in the single-cell RNA-seq adjustment. And yeah, so these are the two main components that we have for our algorithm. So the data, so this algorithm scMerge version 1 was okay-ish to run in 2019 when the number of cells is around with the initial study is around 1,000, but it struggled a little bit in 2023 when now we have like 100,000 to 1 million of cells per study. So we kind of like update our algorithm by considering three main components to address the challenges in this kind of the data structure. So the first one is for the for the multi-layer technical variation. We now consider a hierarchical data merging strategy to so that we can actually merge between the donors within each of the batch first, and then we can merge between the studies. Secondly is that we also consider multi-layer biological variation, where we first if we have like know the phenotypes of the samples, then we can basically build the mutualist cluster within each of the phenotypes and then use to construct to the pseudo replicas. And thirdly is that instead of using all cells to estimate W and alpha, we now construct the pseudo bulk to estimate alpha. So this will because estimate alpha actually include singular value decomposition computation. So we actually if we're using pseudo bulk, that will reduce the time. And this is some of the benchmark results of the in our paper. So we can see that compared to the old scMerge, the scMerge like the scMerge2 without the hierarchical merging, scMerge2 hierarchical will perform better. Also in terms of the scalability, we have improved significantly compared to our old version. And also we are comparable to the other methods that like can output the adjusted matrix, and also is comparable to some of the methods that only output the embedding. And we can also in our paper we show that we can merge 5 million of cells and of the COVID data. Yeah, so scMerge2 is just published in Nature Communications. And so next I will show a little bit on how we can run our scMerge2 using a small data set actually. So the workshop material is on this one, this link. So it's on the Yingxin link GitHub scMerge2 2023. And because I was quite late to decide to using this template to make the material. So currently, if you log into the Galaxy, actually it's showing the scMerge vignette. So if you want to run the scMerge2 2023 material, you need to git clone this one, and then install the scMerge2 2023. But
all the Galaxy ones got scMerge2 written everywhere.
Yeah, but that is a vignette of scMerge.
Oh, okay.
Yeah, it's a vignette, but I add some components in the material, but it's fine if you just look at that. The 80% of the materials are the same. Okay. I actually need to do this myself here too. Yeah, so so this is a very simple data set, I have to say. But the idea is to get people have an idea of like what kind of the data, what kind of the analytic strategy you can think about when you want to integrate the data, and what kind of the parameters you need to like be careful when running scMerge2. So the data set that we are using is from package of scMerge first. So if you loaded this data set and then yeah, so it's a very small data set. It's like 200 cells and then 1,000 genes. So this one is basically within the scMerge as an example data to satisfy the bioconductor requirements. Yeah, and then if you look at the data, so we can see that we have 3 cell types and then 2 batches. So the most default version of scMerge2, as I said, is the unsupervised version. So basically, we will do the graph clustering within each of the batch, and what it needs to be input is the batch information and also the negative controlled genes information. So how do we decide the negative control genes is so this one is provided in the scMerge package. So this is the list that we curated based on some invariant stem cell data set. And because if we look at this data set, it actually is under the ensemble gene ID. So that's why we need to load this negative control gene list. And then because this is a mouse data set, so we need to go to the mouse list. And then this is the single cell stable expressed genes we use. And so with this, and then the input data is the log count matrix of the single cell data set. And then if we have all this, we can then basically running.
For what species do you have the SAG list, the control?
The control.
So here you're showing for mouse, right?
Yes. So you can see.
What else?
So we also have. So for this one, we have human and mouse. And for mouse, we have. So for each one, we have 3 lists. So first is the one that curated from the microarray housekeeping genes, and second one is curated from the RNA-seq, and our one is from the single cell data set.
And same for human, right?
Yes, same for human. So we also have.
What does SEG stand for?
Stably expressed genes. Yeah. So we also have this one, that list. So this it would be the gene symbol. So if you go to mouse again, yeah, so it would be the gene symbol. So basically the same thing, but we just provided 2 data lists for convenience. And then we can check out the results. So now if we look at the points that with different shapes, we can see that it probably means better. And then the cell types that are from the cells from the same cell types are now closer to each other. Yeah, so this is the default session. And next is that if I happen to know some cell types, happen to know all the cell types in each of the batch, using some other clustering algorithm or using the classification cell types, like cell type classification algorithm, then scMerge2 allows you to input this as the cell type information. So now you can just basically input in the cell types, and the thing is that we will still match between the cluster. So we won't force all the cell types that are labeling the same thing to be together. So in fact we're still trying to do the matching. So it's okay if you your this cell type information is like 1, 2, 3, 4 in batch 1, but then A, B, C, D in batch 2. We were still trying to match whether A is 1 or A is B. Yeah, so this will be so this will save time to doing the clustering, but then it will still do the matching. So yeah, this one will be look similar with the one that we just saw. So
Question, so the difference there is with the previous one, it's doing its own clustering,
yeah,
and here you're essentially giving it the clusters,
yes,
but it doesn't care what the description of the cluster is.
yeah, no,
It's just a partition of the cells into clusters.
yes.
Okay.
But in terms of performance, what's better to like give cell type or just do it on the unsupervised?
So it's depending on whether you like my own clustering algorithm, but my own clustering algorithm is just the sharedness neighbors plus the low-end clustering. So if you think this is not good for my data set, not good for this batch, then I think the one that guided will be better.
Maybe I'm missing something, but there is not a circularity in the sense that you find a common embedding, then to classify cells, cluster and label, and how they find the cluster before, then integrating and then finding the clusters again, supposedly influence each other.
Yeah, this is like a very good question. I think it's where like determine like what questions you want to answer for your analysis. So if you want to like still want to identify whether each of the whether like the batch share like common cell types or distinct cell types, I think you can still see in after integration. So this is actually to guide it to integrate the data and correct the batch effects by looking for similar biological variation across the batches. So it's not forcing the cluster to be together. Yeah, so and then I will then go through some of the parameters that can be turned in scMerge2. So first is that how many number of pseudobulk that we want to construct within each of the constraint, like the cell grouping. So currently we use the 30 as default, but we can actually make this number bigger. But then the thing is that if we increase this number, then the number of the computational time will increase as well. And in our experiment, we don't find this will impact our performance very huge. So it's around 30 to 50. Like it's around 30 is actually good enough for the large data set. And another trick that you can do for scMerge2, because it actually returns the full matrix, the adjusted full matrix of the data set. If you return the whole big matrix, sometimes we will actually takes a longer computational time, as well as it will also eat up a lot of memory. And so we actually implement a matrix called get adjusted matrix. So we can actually do it using a for loop, or you can even do it using in parallel to get the adjusted matrix after you estimate the scMerge2 parameters. So this is by first we need to set the return matrix equal to false. And then we need to check. So this is the because currently scMerge2 actually do the cosine normalization by default. So we need to estimate the adjusted mean for each of the genes under the cosine normalization matrix. And then using this information, we can then actually get the adjusted matrix by batch. So this is to do it for within each of the batch. And this will be very helpful if you are doing a very large scale data integration. And also you can also select a subset of genes to return the matrix, and this will also save up the memory in reality. Yeah. So next is the newer part of scMerge compared to the old version is that we actually provide a hierarchical version of it, and it is implemented as scMerge2H. So you can basically set up your own merging strategy to integrate the dataset. And so for illustration purpose here, I will create some fake sample information of this dataset. And then so for now, for each of the batch, we have 2 samples. So to perform scMerge2H, we need to create 2 information. So one is a hierarchical index list that indicates the indices of the cells that are going into merging. And second is a batch information list that indicates the batch information of each merging. So I will show you how to construct these 2 lists. So first is for the hierarchical index list. So I will show you how it looks like. So basically we have 2 levels. So each list here will indicate like level 1. How are we going to do the merging in level 1 of the hierarchical merging, and level 2 indicate how are we going to do the individual level 2 individual matching. And this is showing that in level 1 we are going to perform the hierarchical merging within 2 batches. So it's batch 2 here, and then batch 3 here within. So this is 2 merging. And we can jump and to see the batch list, how it looks like in the batch level 1. So this is indicating on level 1 within this batch 2. So suppose we have sample effect. So this is the sample information. So this is sample 1 and sample 2. So on the level 1 within batch 2, we will do the correction between 1 and 2. So this is giving the batch information. And we can then go back to the level 2 and see. So this is indicating like level 2, we will only perform 1 data correction, and this includes all the cells here. So it's because we have 200 cells here, so it's like 1 to 200. And then if you look at how the batch so if you look at the level 2, yeah, so this is indicating now on level 2, we are doing the batch correction between batch 2 and batch 3. So with this information, and then we can input this one into the batch list and the hierarchical index list into the hierarchical scMerge2 function. And one thing that I want to highlight here is that the number of unwanted variations that we want to remove. So this is a factor indicating the number of unwanted variation we want to remove from the data set. And by default, we set it as 20. But in the hierarchical system, because we are actually doing the batch correction twice for some of the samples, so I find that sometimes the first level need to have a smaller unwanted variation to be adjusted, and then in the second level we will set a larger one, like 5 to 5 or 10 in this case. Yep. So this is run it please. Yeah, it will take a slightly longer time than the one that without the hierarchical, because we are performing multiple correction.
Does it support an arbitrary number of levels?
So we actually, yeah, it's very flexible. So you can do 10. Yeah.
What's the performance at certain amount of depth? Are you going to get really weird behavior? It goes level.
Yeah. So if you go do 10, then it will be like overcorrected.
Right.
Yeah. So I'll like my strategy when I'm doing the data integration is I still I will actually look within each of the batch to see whether there are like sample, donor, or like cohort effect within each of the batch or studies. And then I will for this kind of the study, I will do this hierarchical thing. But then it's not like for all the studies. And then the output of this is actually it will be a list of the adjusted matrix. So this is indicating the first level of adjustment, and then this is the second level adjustment. And we can then still input this in our single cell experiment, and then we can look at how it looks like. So yeah, so because this is like I created a fake sample, so it probably is not necessarily better than I didn't do this hierarchical things. So as I just mentioned, this can be a very flexible merging strategy. So we don't actually need to do it for all the indices on each of the level. So here I illustrate that like if we remove this in the first level, now we only do the adjustment within batch 3. So we don't do anything for the batch 2. We can also run scMerge2. So now we only have level 1. We have the batch 3 information here. So we will skip batch 2 in our integration. And yeah, so this is just running it again.
While it's running, I have a question about something earlier. When you provide your own clusters to it, can you specify that certain cells are not in any cluster, like give them an NA cluster, and then have it not use those in the?
Oh, this is a very good question. Currently, we don't have that.
Because I know there are some clustering methods that take the sort of the marginal cells and don't put them in any cluster.
Yeah, so yeah, this is very good things that we can actually implement this.
And so
Yeah, but
it would be good to like take those cells along for the ride, but not include them in the cluster matching.
actually in the old scMerge version, we have that function, but I didn't implement it in the newer version. I was a little bit lazy. Yeah, but in theory, if that cluster doesn't look like anything else in the other batch, it won't be matched to the it won't like be the mutualist cluster. So it will still like I guess scMerge2 will still do a right thing. Yeah. Oh, that's right. Yeah. So next I will have another example to illustrate how we can using this phenotype specific pseudo replicas. And we will focus on the size-seq data as an example. So, oh, I didn't actually install this.
I'll ask you later. So if your cell types, or can you do like a cell type call? If you have cell types that are capturing technical artifacts in one sample, the same technical artifacts in another sample, will those things actually end up getting removed? dashboard. So that's a nice solution.
That's a very good question.
Just skip updates.
I would imagine in this case, the negative control genes would be the one that try to estimate the unwanted variation. But if it's not successful, then you will have unwanted variation cluster in your merge dataset. Yeah, so the algorithm have like two components trying to do the estimation of unwanted variation, and they do it jointly. So if one fail, and hopefully another one will come and save the situation, but not necessarily all the time. So I think it's a very data dependent.
So you're better off doing a lower resolution cluster.
Actually, a higher resolution clustering will be better for not overcorrection. Because
Higher resolution meaning more clusters.
More clusters. Because so scMerge2 is relatively
No.
yeah, so scMerge2 is relatively less sensitive to the clustering algorithm because we are using the pseudo bulk algorithm. Yeah. So now. Then I do this. Yeah, so this is 3,000 cells examples, and this is from COVID-19 studies, and I have subset the data set that now if we look at the data, we have data from two conditions, so healthy and mild and moderate symptoms, and then we have two batches, so one is the Cambridge, another one is NCL. And so I will skip to the size-seq example. So this is a size-seq data set. So we have the protein abundance data in the alternative expression matrix called ADT in this single-cell experiment object. And if we go look at, so this have 192 proteins, and we can look at the data set. To see the batch effects that exist in the data. So, or we can go to scale to see it. Yeah. So this is the size-seq data set. So we have cell types that are from so this is a quite well annotated cell types, and we have two cohorts. And if we color the cohort, color the data with this Cambridge and NCL, we can see it has a very strong batch effect. And also there is the condition, like the severity, like the condition phenotype information within each of the batch. You can see that they are actually quite separate to each other. So then, so there are a few things that we need to actually take care when running the data, like protein abundance or site-off compared to the single-cell RNA-seq data set. Because first is that the negative control genes. So for RNA in general, we found that around 1,000 negative control genes will work good in general. But then for the protein abundance and also site-off data, we actually found that we still need a reasonable large number of negative control features. And so we actually found that using all proteins in the data set as negative control genes will work well. And secondly is that because now our number of features are reduced from 10,000 to only 200. So the IUV, the number of unwanted variation also need to reduce accordingly. So usually for side top data, when we only have 40 to 50 features, then I will usually start from 5 and then add it to 10 if there are more unwanted variation I want to adjust. And for size 6, this one have 200 protein, so then I will do the 10 here. Yeah, and then compared to the previous one that because now we have the phenotype information, so we just need to input to the phenotype information into the condition, and yeah, and then we can just run it. Yeah, so maybe we just go to this. Yep, and then this is after the merging. So we can see that now the batch batch kind of like mix better than before. And we still can see the clear condition effect between the samples that are from different like phenotypes. Yeah, and then this is basically like the end of the workshop. And any questions?
I have a curiosity question while people are processing and thinking of questions. So you have stably expressed genes. For human, it looks like these are ribosomal proteins, spliceosome, DNA replication. I notice you don't have histone proteins. Do you know why they do not show up in your list? I'm just curious because a lot of times people think of them as pretty stably expressed.
I see. This is a good question. I have very limited knowledge of these genes.
And I may be totally wrong, that I've heard people say like their histone proteins are highly expressed and cells need them to divide, and I may be wrong, like maybe so your empirically derived list
Yeah, so
has ribosomal proteins and spliceosome for sure.
Yeah, and then we are based on a single cell data set of like a SMART-seq data. Are these genes also quite like abundant in the cell?
So they should be in any cell
I see.
to make the nucleosomes. And I've heard that they're used as housekeeping tubes, but anyway, maybe there's some reason why they are not.
Is that for bulk or single cell?
I mean, any cell has histones, right?
Well, but you have a different number of histones if you're in G1 or G2, right?
Maybe that's why it's not.
So it could be a cell cycle thing, but as long as you're in bulk, then the cell cycle effect is not an issue. So maybe it's only good for bulk.
Yeah.
That's just a guess. I have no evidence for that.
Yeah.
Yeah, I need to go back to check that data set. It could be they are all by model, so probably is they have a higher variance.
That's a pretty good explanation. Any questions?
That's why I got the biology degree too.
Yeah, I don't have a biology, so that's probably the reason.
Let me check online. I've been checking, but they've been quiet.
So, hopefully I'm expert in it. So in REV, the iteration across number of k usually needs to be checked somehow. Yeah, so how your data set get correct as you get k. If you get very large number of k, your biology starts to be corrected as well. Do you have a strategy to iterate and to suggest to the user how to do it manually or not?
Yeah, so usually, so if the replicas are estimated perfectly. So this number will be quite robust of a default number. So it's 20. So after this, it won't have a huge impact in the in the normalization. So of course it can't be too large for the RNA data set.
This is the K of 20.
The K equal to 20.
So you're saying that there are 20 factors you are trying to correct. 20.
Yeah. And yeah, so usually I would do it by like the common matches in the data integration. So it's like and then yeah, so it's to look at whether this and then another strategy it actually is to look at the housekeeping genes expression after adjustment. So if you before the adjustment, you will see the gene expression of from the housekeeping genes from each of the batch will have a very different distribution. So supposedly after your batch correction, if you successfully remove all the unwanted variation within each across the batch, this distribution will be similar. And so this is one thing to look at whether the batch are corrected. And secondly is that we want to see whether our cell types that we are actually like overcorrected. So then it will be like so one thing you can virtually look at it in the UMAP plot whether they are merged with some other cells like totally is like a big cluster now. Yeah, and this is another way to like guarantee your biological signals are not overcorrected. Yeah, and you can also like by looking at the certain cell type markers, but then you need to actually know this cluster, like is CD8 T cells, and then I go to the CD8 T cells, and then I whether see this marker is still the gene expression is higher, the CD8A is higher than the other cell types markers. Yeah, so this is for like scMerge, I would recommend to look at the gene expression to check the level of the adjustment.
I'm sorry if you mentioned this before, but you're using these are all individual summarized experiment objects that you're using these crossway hierarchical and clustering analyses to break down from dimensionality. So on the back end, it's just a bunch of matrices that you're going through. I'm curious, have you ever tried doing this on an actual single cell analyzing this performance or something on that scale?
So you mean doing a large scale?
Like if you're trying to use a data set of 50 different PBMs, like PBMC pages, and have you tried replicating that like 50 times, then you see the same performance going through these merges?
She does a 20 data set thing with whatever, how many million cells.
Oh yeah.
That's atlas scale, I guess.
Yeah, so actually in this case, this set of the code will be very important. So in this case, because it's so if I do it on the whole matrices and I do the adjustment of the because we are actually doing the W matrix times alpha matrix. So if we doing on the 500 million or 5 million, no, 5 million cells, then it will be quite problematic. So yeah, it's not problematic, it's just slow and it will eat up a lot of memories.
Yeah. So
yeah, so if you I usually would just do it by batches, and even you can split it to like 100,000 cells each time. So this is just do the adjustment at the end. So it's just this part. Where is it? So is that after we estimate the alpha, so this part is just to do the W times alpha and then Y plus this operation. And this actually is the one that will blow up the memory part. So the idea is that I will usually do this part by batches, and then each of the time I have the adjusted matrix, I will save it to the H5 file.
Oh, so I'm thinking that if you that since spatial has laid arrays of what going through right there, you probably not have to do these batches right there because then okay, it's just that if you did straight batches, of course you would blow up your memory right there, but since you're doing it by batches, that explains that right here on this. Okay, like
Yeah, but the estimation is still the whole. Need to use all.
right, but you're using a portion of it at a time, and then completing it after the rest, and then putting it going through right there. It's just that I was concerned how are you able to do this on because when I ran this on with the old algorithm, I couldn't get it to go through memory. I was interested in optimization to reviews right there, but that explains it. But, yeah.
So if different set types have different effects on batch, let's say, as it's often the case, does this algorithm allows for different batch effects based on the cell type, or assumes that all cells are affected in the same way by batch, let's say?
It's not assuming different. It's not assuming the different cell types have the same batch effect, and this is probably that why we need a certain number of unwanted variation, because each factor could estimate different types of the unwanted variations of different cell types.
Okay. So one component could be just some effect on T cells.
Any last questions? If not, it's next speaker.
Can you